name is Dr. Chris Jenkins, and I am the CEO of the Orianne Society and the host of the Snake Talk podcast, the podcast where you learn about nature's most feared, maligned, and persecuted animals. I invite you to listen to this conversation, and maybe you'll find that what you perceive as fear is actually rooted in a deep fascination. Welcome to the Snake Talk podcast. We are continuing our student perspectives series, and I wanted to do one of these episodes talking with some undergraduates. To date, we've been talking mostly with graduate students, but I am happy to have both Cameron and Ariana, from the Department of Biological Sciences at University of Northern Colorado. Cameron just recently graduated, and Ariana is still undergrad. So excited to talk about what they have going on. Welcome to both of you. Welcome to the podcast.
Yes, thank you so much.
Well, as you may know, I like to start from the beginning, and I'll start with you, Cameron. We'll get into what you guys have been working on in your undergraduate, but you are involved with Venom-related research that, we've had some previous guests, your advisor, fellow students from the same lab, but snakes, venomous snakes, venom, these are all things that are really outside the box for most of society. So how did you end up getting into this, Cameron? Was this something were you always into snakes, or is this something that you kind of picked up later in life?
Yeah, not necessarily. I've always been interested in just nature in general and animals and just being outside. But particularly this lab kind of was just a bit random, I guess, because as part of our program, we need to do a capstone and doing some sort of research. So I was just looking through all of the labs available, and I saw Dr. Mackessy's lab that obviously worked with venom and snakes and stuff. And specifically in his description, there was a lot of things that kind of mounted my interest. Like there was healthcare involved. There was like ecological evolutionary concepts, just so much stuff that you could really use venom for that I didn't really know. And at this point, I didn't really know what I wanted to do with my career, with my degree. So I thought this would be a good point just to kind of have all these options at my fingertips. And yeah, I just kind of got involved with this, started doing some simple assays and just taking care of the snakes. And I just kind of loved it ever since. And yeah, now I'm really involved with Ariana. Ariana and I have our own project that we're working on. So, but yeah, it was kind of just a bit random, I guess.
Yeah. So you were interested in nature, and did you grow up there in Colorado?
I actually grew up in a small town in Michigan called Grayling. It's like 3 hours north of Detroit.
Yeah, I know where it is.
So yeah, really.
Yeah, I mean, I haven't been there, but I know where it is on the map. Yeah, so
yeah.
Excellent. So you grew up in a relatively rural area, close to some pretty wild country, and moved to Colorado, and we're into nature, and then just kind of came into it since. But you said you're taking care of snakes, and I know in Dr. Mackessy's lab, those would be venomous snakes, many of them, most of them probably, and so you've obviously taken to it. That's pretty interesting. So you've just finished your undergraduate. So you're in this major, biology major, and you mentioned interest in health sciences. Mm-hmm. Were you kind of focused, or are you still focused in human health, or has this whole experience with snakes kind of brought you more to like that evolutionary ecology side? What's kind of your interest in your undergrad major?
Yeah, this field is really interesting because I started off as having an emphasis in ecology, evolutionary biology, and I really thought that's where I wanted to go. I wanted to work outside, work with these animals in their natural environments. But this field has been very interesting because it really incorporates the evolutionary side, really impacts the health side as well, which is what I really love about our current research, because, having this diversity of snakes and snake venom, it really impacts the symptoms we observe from snakebite from individual to individual and population to population and so on. So it's this really cool mesh of both kind of fields. And I really have taken to that. And it's kind of made me think of how I want to include this in my career in the future, of kind of developing better antivenoms and how we can take care of people that have been bitten by snakes.
Yeah, that's great. So how did you end up from Grayling, Michigan? How did you end up in, was it Greeley? Is that where University of North Colorado is?
Yeah, Greeley. I know Greeley isn't the closest to the mountains. It's about 45 minutes away, but I've always, like I said, I've always been involved with nature and love the outdoors. So I thought it'd be a good opportunity just to explore while I was young and study out here. And it's been a great time. I've had a great opportunity of getting to the mountains and having some really cool camping experiences and stuff like that. So
excellent. Well, Ariana, how did you end up getting into snakes and venom and all of this? Were you one of these children who was just crazy about snakes, or again, like Cameron, was it something more later in life for you?
Yeah, so I actually grew up really loving snakes and reptiles in general. I got really into snakes when there was this one time my cat brought a garter snake in, and 9-year-old me, obviously, I'm like, I'm gonna take care of this snake, and that's where it all started. And so when I was in my freshman year here at UNC, that would be fall of 2023. Some of the professors who had labs came in and talked to my Bio 110. Mm-hmm. And, I just immediately was like, man, research sounds really cool. And what do you mean I can look at snakes? Yeah. So it was a really big deal. I got very excited.
So did you grow up in Colorado then, or?
Yeah. I grew up between Castle Rock, Colorado, and this tiny town in Kansas called Osborne. It's like an hour outside of Hayes. So I grew up really between those two places.
So you grew up out in the prairie then, really?
Oh yeah, we have a farm and everything. We'd find, rattlesnakes everywhere. It was always a really cool thing to me.
Yeah. Okay. So what was the decision to go to Northern Colorado? It sounds like it wasn't for snakes specifically. That came, through the experience you had with Dr. Mackessy, but was it just that was the close university out in the prairies of Colorado, or what was it when you finished high school? Did you go there to become a biologist? How did that transition from high school to college work for you?
Yeah, so I had my entire heart set on going to Kansas State University, but even more than that, I had my heart set on majoring in biology. Biology has always been my goal my entire life. I was, insistent on being an epidemiologist when I was a kid, but we went more towards the snake route. And honestly, UNC is a great place for sciences. It really is, much more than K-State was. It's more of an ag school. And the, affordability was a huge reason too. Yeah. And it's very close to my home. I'm like an hour and a half away. Yeah.
Those are all important things for people to think about as they're moving on. When you're thinking about going to college or university, affordability is a huge thing, especially these days. It's insane how much some of these universities cost, and so just going in state saves you quite a bit of money. And then some people like to be relatively close to home. Some people want to be relatively far from home. So, but just however, taking those types of things into consideration can be really important. So you're at University of Northern Colorado, and you have this kind of experience where you meet Dr. Mackessy, but you were already a biology major. This just kind of like honed in on kind of where you were going to focus. Is that fair to say?
Absolutely. It's a little bit funny. I had the exact opposite path of Cameron. When I came here to UNC, I was focused on the medical side. Mm-hmm. My major was in biomedical sciences. And I specifically wanted an experience that wasn't just going to be medically related, because I love all of biology. I could have an interest in every section. And so I wanted something a little bit more outside of the medical route, but could still, be a little bit related. And I fell in love with this lab so much that then I changed my major to ecology and evolutionary sciences.
Okay. That's a great story. Like I said, pretty contrasting stories there, but you guys have landed in similar places. So let's talk a little bit about University of Northern Colorado relative to, ecology, herpetology, snakes. Obviously, Dr. Mackessy's there and, has had quite a career. But do you think University of Northern Colorado, this is kind of a question for either or both of you, do you think it provides a lot of resources for somebody who's interested in getting an undergraduate, that's, say, has herpetology as a component or snake-related things as a component?
I think so. UNC is quite small, I guess, relative to at least some of the other like schools around here, like CSU or Boulder. And I think that really, even though it is small, people may think that there might be less resources for the students to use. But I think that it really is a benefit for us because we really get to have this more kind of intimate relationship with our professors and just have this more hands-on experience like we have had with Dr. Mackessy. And I don't think people at other schools really can say the same thing because the professors are so busy with their own stuff and there's so many students that they don't really have enough time to dedicate the time that we've gotten with Dr. Mackessy. So it has been great for both of us, I think, just being able to get so involved because there is less students essentially competing for the same thing.
There's definitely something to be said for that. These bigger schools, they might have more resources in some ways, but you also can get lost very easily in the crowd, and there's less opportunities for the type of interaction like you guys have had as undergraduates. So that's excellent. So curious for both of you, how we don't need to go into depth yet. We're going to get there, but on what you've been doing in Dr. Mackessy's lab, how did you get to that point? Meaning any undergrad at any college or university can go to their classes, do well, pass their classes, and get their degree and move on. But obviously you guys have gone a little bit above and beyond. And so I'm curious that story for both of you. How did you get where you were like to Dr. Mackessy's lab and you were able as undergraduates to do to work in the lab? I think, Cameron, you mentioned something about a capstone project or something like that, but I'm just curious if both of you could kind of tell us that story.
Yeah. So I actually did not even know that we needed a capstone project when I first asked Mackessy to take me on. I just emailed him. I was like, hey, the research that you're doing is so interesting. Is there anything I can do to get involved? What are my next steps? And, I was sent a lot of papers to read of his research that he's done. And sitting through and reading that, I did not understand anything that was in them as a freshman with all the technical lingo and all of that. But I still really, really wanted to try. And so after I read those and I kept emailing with him and I met with him, Dr. Mackessy was like, okay, yeah, you can come to our lab meetings. Just show up. We'll start talking about things. You can maybe help with one of the grad students. And so really for that first whole semester, since I really only joined halfway through that semester.
This was your freshman year that you made the connection?
Yeah.
Okay.
Exactly. My fall semester, first semester, my freshman year. And so I just sat in the lab meetings for a while. And I slowly started to learn lingo. I started to learn, what does this mean? What are we actually looking at? All of that. So it was really helpful to have that one semester. And yeah, in that second semester, so spring of 2024, I was then not the only undergraduate. And there was 4 undergraduates in that lab, including me and Cameron. And Mackessy started really working with us to try and develop something that we could start working on together.
Okay.
If you want to talk about it.
So how did you, I want to get into that, but I want to hear the story, Cameron, how you got, like you could just go to undergrad there, get your grades and graduate and move on. But how did you get more linked to a particular lab?
Yeah, I think it was kind of a funny story about the 4 undergrads, not the cleanest projects, but I think I was just because you don't really know exactly what a lab has to offer until you've been in it for a while. So I thought that if I did get into it relatively earlier, probably earlier than most. Ari was an exception to that where she joined her first year. But I thought that if I got in early, I'd be able to kind of get to know the people that are in the lab, maybe get involved into a more intense kind of research project that I was interested in, not just kind of doing like the surface level kind of stuff. So I thought that if I just kind of stuck with it, I would be able to learn all of the terminology, just like Ari, and like learn more about venom and snakes in general, which is something I really didn't have a lot of background in. So I'm always down to learn kind of about a new subject, and I kind of just stuck with it. And yeah, we're here now.
Yeah. Okay, great. So you mentioned taking care of the snakes. I'm assuming, Ariana, you take care of them as well?
Oh, yeah.
So tell us a little bit about the snakes. Like, assuming you have a pretty big snake collection there, like just tell us about some of the species that you guys work with in captivity day to day.
Yeah. So in our multiple snake rooms, we have around, I think, 165 snakes. Not all different species, but the majority of them are rattlesnakes. That is what our lab works with most, is those rattlesnakes. We have a few non-venomous species, and we have some from other places. Like we have, a gaboon that we work with, some other arboreal species. But yeah, it's mostly our rattlesnakes that we're working with in those rooms.
And are those prairies, or are you working, I'm assuming you're working with a lot of western diamondbacks?
Yeah, there are a lot of different species. Oh, I don't actually know how many different species we have.
Yeah, there's quite a few. Like we have some, I'm actually more familiar with like the Latin names than the common names. We have like some Cerberus, some western diamondbacks, viridis, several subspecies as well. So we have some like lutosis, some sidewinder rattlesnakes, some cerastes. So we have a lot of rattlesnakes from just in and around the U.S. that we work with and that we take care of.
Gotcha. Do you have any timber rattlesnakes or?
Oh yeah.
Yeah. I think we have one.
Yeah, big one.
Yeah.
Big one. Yeah. Huh. From out that way or from further east, or do you know?
I don't actually know.
Yeah, we're not exactly sure.
I'm sure it says in the sheet somewhere.
Yeah.
Huh. Okay. Interesting. I did my PhD work on lutosis many years ago. But yeah, so you got 165 individual snakes across multiple species, most of them venomous. I'm assuming a lot of the care for those falls on this team of 4 undergraduates. Maybe the graduate students help as well. But, what does that entail, maintaining such a large snake collection? Just talk about like day to day. Like, what do you guys have to do to like keep 165 snakes going?
Yeah, so we, every single semester, at the very beginning, sit down with the whole group and figure out what times everyone's available. Kind of, are there times where there's going to be at least like a group of 2 or 3, including the graduate students. The graduate students, there always has to be one down there with us if the undergrads are going to be down there. And 2 to 3 times a week, we're down there for typically about 2 hours at a time, cleaning out water bowls, cleaning out the cages, doing feedings, all of that with the graduates. And yeah, it's about how we keep it up. We just, every few days, we're down there for 2 hours just working.
Gotcha. And you guys enjoy handling the venomous snakes, I'm assuming?
Yeah.
We've learned a lot of techniques on handling.
Yeah.
A lot of training.
Yeah.
It's funny because at the start, obviously, sorry, go ahead.
I was just going to say, and I'm assuming you guys actually, I handle a lot of venomous snakes as well, but I typically tubing them. I'm assuming you guys are actually, where you're working with venom and you need to get venom, you're actually handling these animals where you're holding them by the head and getting venom out of them. Is that true?
Yeah, so mostly we're just, if we are going to handle them at all, we just use 1 or 2 hooks and we put them into a bucket temporarily while we clean their cage.
Gotcha.
And it's quite the privilege because at the start, we start slow, just working with the non-venomous stuff. But then as we get a little bit more comfortable, we can work with the venomous and then even like the bigger venomous snakes that we have. But the venom extractions mostly lie on our professor, so Dr. Mackessy. So he puts them we anesthetize them with isoflurane, and then he is able to pin their head down and then squeeze out their venom. We're not as involved with the extraction part as he is, because obviously there's a liability there.
And so you guys are able to, you're handling snakes in the lab using hooks and tongs and things of that nature, it sounds like. That's great. And have you, during your time there, do you guys get to go out in the field at all and look for snakes, either for the collection or just recreationally or out on projects where you see some of these animals in the wild? Hey everybody, I just wanted to take a break to let you know that you can stay connected to the Orianne Society by signing up for our monthly newsletter. Be the first to receive exciting updates on reptile and amphibian conservation, from the latest news and staff blog posts to exclusive announcements like upcoming Snake Talk podcast guests. Go to our website at www.orianne.org and sign up for the newsletter at the bottom of our homepage.
Yeah, absolutely. There's different areas that we go where we have very commonly found, typically, prairies? Prairie rattlesnakes. We go out, we collect as many as we can, and as many as we can range from, a day of getting 0 to, I think, a day of getting upwards of 150 at one point, or 30s typically. And those ones, when we're doing specifically that field work, we bring back to our lab. We do venom extractions, usually a whole day, and then we release them again. So we've gotten a lot of field work done where we get to actually go out, find these snakes, catch them, bring them back, which is always fun because you never know what snakes you'll find. You could find these prairie rattlesnakes. You could find some milk snakes. You could find racers. It's a fun time.
Yeah, sounds like it. Okay, so let's get into the undergraduate research that you guys have been working together on. And it sounds like all of this team of 4 undergraduates have been working on. So first of all, so you're helping out in the lab, you're cleaning snake poop and, but getting the opportunities to sit in on the lab meetings and interact with the grad students, and you're just kind of getting immersed into all of this. But the idea of undergraduate research comes up. So is this, how much did Dr. Mackessy and other members, whatever, grad students, postdocs, whatever they might be, did they really develop the ideas for this? Or how much involvement basically did you guys, this team of 4, how much involvement did you have in actually kind of designing the project? Or was your primary role kind of implementing the project that they developed?
Yeah, it definitely started off as more of just direction from Dr. Mackessy and some of the grad students. As of now, it's only Ari and I working on the project. So the very beginning, our professor picked out 39 rattlesnake venoms from across the Americas. So from the U.S. down to, southern of the South America. Our first goal was just to kind of characterize these venoms. So we have a few kind of classic biochemical assays for PLA2s, SVMPs, LAAOs. So the first kind of idea was just to get us familiar with these common biochemical assays that we could use in the future.
So in more let's say you go back to Grayling and you're sitting down with your grandmother and you're telling her what a biochemical assay is, how would you describe it?
Yeah. Honestly, in very simple terms, we are kind of, breaking apart the venom, looking at exactly what's inside of it and how much of that is what's inside of it. So the different proteins that are in snake venom, we can take these experiments and really look at how much of that is in there and what that does within the venom.
So that's what these assays are telling you. So that's kind of the first part of your project, that you have this wide range of rattlesnakes, really across the range of rattlesnakes, and you're basically kind of describing the composition of their venom. Is that fair to say?
Exactly.
Okay, so that's one part of it. And how, where are you guys in that project? Is the type of thing where you've completed an incredible amount of work for all these species. You've done it for 2 species. You've done it for one type of protein across all of them. But are there any kind of results that you're already seeing, maybe about the diversity of venoms across rattlesnakes or anything like that? Just kind of what have you been figuring out with that first step of the research project?
Yeah, absolutely. So with that first step, we really just wanted to do a very broad look, right? We wanted to show that these rattlesnakes, despite, all being rattlesnakes, have very wide compositions of venom. They're very diverse. And so for that time when it was a group of 4, that's what we did. We looked at that wide variety. We were just showing that diversity is there. And that quickly then evolved into, we have this diversity. We can see these proteins. We've described them. What can we do with that? And that's then when our team of 4 turned into the team of 2. And Cameron and I started looking into more specifically the proteins within them. And that then turned into also looking into antivenoms with those proteins.
So before we leave the first part, you talked about incredible diversity of proteins and venoms and so on across species and so on. How about is there any finer level of diversity? So are you looking for example, within a wide-ranging rattlesnake species? It's known with multiple species now that the venom can be different in different parts of the range. Or take some species of rattlesnakes, like if you take a neonate versus an adult, their venom can be. How did you guys incorporate that into this kind of phase 1, descriptive phase? Were you sampling not just across species? Were you also sampling within species, across age classes? Just speak to a little finer detail there if you could.
Sure. Yeah. So for this project, it was very broad. So for this one, it was mostly between species. And then there was 2 samples that were from the prairie rattlesnake, so from Corylus viridis, that we demonstrate that variation. And I believe our professor has talked on here about Kara Smith's work with the prairie rattlesnake, where those in the northern part of it range has a lot of this toxin called myotoxin A. And then right below I-70, we have this switch in phenotype of the venom where it's switching from high myotoxin A to a high SVMP composition. And it's theorized that could be due to kind of prey in the area and probably some other aspects as well. However, I don't know if that's been fully kind of fleshed out yet. But
again, talking to your grandmothers, describe what a myotoxin is, just so people have an idea. How does that affect a human, for example?
Yeah, myotoxin is generally myotoxins just are attacking muscles, and it can cause variations of paralysis. So I believe with myotoxin, it causes a tetanic paralysis, which means that your hands or whatever part of your body is struck by this snake or with this toxin, it's going to be contracted more than it really wants to. So as you can expect, that allows for easier prey acquisition for these snakes.
Yeah.
So generally just attacking muscles with different mechanisms by which they do so.
Yeah. And then you mentioned south of the highway or interstate, whatever it was, that there was another type of venom. You said SV.
MP, yes. So snake venom metalloproteinase.
Okay. Yes. And so describe that in general, again, in terms of the impacts it might have on a human or a prey item. What's it do?
Yeah, so SVMPs or snake venom metalloproteases are generally kind of tissue damaging. They are kind of breaking apart a lot of the body, so these tissues, and kind of allowing for these other toxins to access regions they otherwise wouldn't.
Okay. Interesting.
And again, there's different variations of these proteins. There's different isoforms is what they're called, just kind of different variations of the same protein that does the same kind of general thing, but they have different kind of affinities for certain areas and stuff like that. But generally just kind of tissue damaging.
Yeah. So any other interesting, you mentioned again, staying in this phase 1 team of 4 type time period, you mentioned this kind of interesting thing that you found. Obviously, it sounds like you sampled the prairie rattlesnakes in more depth. So you were looking at within the species, but you could kind of call out this interesting pattern where you saw this variation within the species. Are any other interesting things that from the broader scale work, we found timber rattlesnakes are incredibly neurotoxic, where these species, is there anything that jumps out as almost superlatives that you learned about this real broad scale look at rattlesnake venom across the continents?
Yeah, absolutely. One thing that we really saw, and that is honestly something that a lot of people should know, especially if you're from the Arizona area, is when we're really looking into that, we see a lot of, what is it? We have the Mojave rattlesnake, right? And a lot of people know that because there's a whole toxin family named after Mojave called the Mojave toxin. And a lot of people are like, well, Mojave rattlesnakes are the most dangerous in that area because of that Mojave toxin. But one thing that we can really see within some of the stuff we were looking at is that they're not. And not even in Arizona. The snake that has the most Mojave toxin heavy venom is the tiger rattlesnake within Arizona.
Oh, interesting.
And it was very interesting seeing that because it was like very comparable. And yeah, so that's one thing that we really get to see is like breaking down that venom wise. All those different like misconceptions almost being broken down.
Yeah, interesting. Any other superlatives, or those are a couple of the big ones, the prairies and the...
Yeah, I think those are the biggest ones.
Toxis.
Yeah, one of our biggest, I think one of our most telling assays was we did a gel electrophoresis, so separating the proteins in each venom by molecular weight. Mm-hmm. And after that, we can dye them and really see kind of what classes of toxins are found in each venom. And we can break them down further into having, a lot of SVMPs or like PLA2s, just different kinds of those proteins. But across the board, there's very little consistency between snakes. And then that's only kind of one aspect of venom variation. There's how active are these proteins involved, or how active are these proteins in the venom, mm-hmm. even if there's the same concentration of them between two snakes. One might be better at its job than the other one.
Yeah.
So there's many levels to the variation part.
Gotcha. Interesting. Okay, so phase 1, you move on to phase 2, the team of 2 now, which is the 2 of you guys. And so let's talk a little bit more about that. So what have the 2 of you been working on in your undergraduate research after phase 1, this broad scale work has been completed?
Yeah. So after that very broad scale, again, like we were saying, it dropped down to just the 2 of us. And we really tried to think of something that we could actually dive more into rather than doing just a broad comparison. And at the time, both of us were biomedical sciences majors. And we were like, okay, well, what, medical implications can we actually start looking into? And obviously, when you think of snake bites and medical, we think of antivenom. Mm-hmm. And so we wanted our main focus to then go into antivenom and kind of how that worked. So we had to start learning, how does antivenom actually work? How do we make it? So there was a lot of research into that, trying to understand it better between the 2 of us. And
so how, just real high level, again, you're home talking to grandma, how is antivenom made?
So antivenom is made just in a very, very general look. We take parts of the snake venom, we inject it into an animal, typically a sheep or a horse, and we then extract the plasma from that sheep or horse and use the antibodies that it made to fight it off and make those antibodies into the antivenom.
So keep going with what you guys are doing. So you're entering phase 2, you're interested in antivenom and how kind of the broad data set that you collected informs the antivenom, or were you collecting a whole new data set? How did that come together?
Yeah, it's so, like I mentioned earlier, it's this really interesting kind of meshwork of ecology, evolution, and then antivenom production as well, because as you can imagine, not one snake bite is going to enact the same effects. We can't just have a single antibody against all snake venom. So as we've been talking about, like these SVMPs, PLA2s, they're all conserved within rattlesnakes. So all of them express some form of these proteins. And that's kind of what we're targeting with antivenom, something that will work against all rattlesnakes and not just 1 or 2. The 2 antivenoms that are available for use in the U.S. right now, Crofab and Antivip, they're approved for all snake bites within the U.S. So to do so, we asked the question, like, how well are these antivenoms actually binding to the toxins found in all these venoms? We see that they're incredibly diverse. Are they actually able to to bind to these toxins, considering that diversity. So that was kind of our first step there.
And what'd you find? Or are you guys still in process?
Yeah, so this was one of our first steps in kind of analyzing these antivenoms. So we did a Western blot. And what we do is we take our gels that we described earlier and we put them on a sheet of paper. And then we administer a few kind of fluids onto them. But after like the third or so fluid we put on there, we put some antivenom on there. And then that antivenom has a sort of color attached to it. So when it binds to the proteins on that paper, it'll kind of show purple, which we can see that the antibodies and the antivenom are actually binding to the toxins. We find that the results between the gels and the Western blots are very consistent. So we're seeing that the antivenoms are able to bind to a lot of these conserved toxin families, except for one, which we find interesting. Maybe Ari wants to talk about.
Yeah. So as he was saying, we will see this bright purple where we're seeing the binding.
Okay.
So anywhere that the antibodies within the antivenom is attaching to the toxins in the venom, there's going to be that bright purple. Mm-hmm. But there was one whole toxin family, which was within myotoxins, where we just weren't seeing anything. There was nothing showing up, which was interesting because there have been past papers that show that binding. And so that kind of shifted our research a little bit into trying to look into that, where we tried a bunch of different things. We did that same process of this Western blot with only myotoxin, where we got binding. It was a very interesting, 2 months of us trying to figure out what happened there. And throughout that process, we were presenting our poster at some of our school things, all of this. And a couple people brought up some very interesting questions talking about, obviously it's binding here. Like, even if it's not like throwing the myotoxin aside, everything else, it's binding. But binding does not equal neutralization. So the antivenom could be binding onto those toxins, but it doesn't mean it's stopping them from causing damage within a body, right? And so after trying a bunch of things with our myotoxin, which is still something we're working on, trying to figure out what happened there, we really started to think about, what does this neutralization look like against this binding? So that's where all of our research is focused on right now, is how well it's actually neutralizing.
So they're binding, you're seeing this binding, but you were assuming that binding means it's neutralizing the effects of that protein or the toxins, but that's not necessarily the case. And so is that not the case for many of the different proteins, or is it just that myotoxin or particular proteins that it seems not to be neutralizing? Or do you know yet?
Hey everybody, I just wanted to take a quick break and tell you that the Oriana Society is dedicated to protecting some of North America's most at-risk species and their habitats, like eastern indigo snakes in the longleaf savannas, eastern hellbenders in the Appalachian highlands, and wood turtles in the great northern forests. Healthy ecosystems depend on these incredible animals. But these landscapes and species need our help to survive. If you care about reptiles, amphibians, and the wild places they call home, visit www.orian.org to learn more about our work and how you can get involved.
So we kind of assumed at first that binding did equal neutralization, but we just don't know for sure.
Yeah.
So we wanted to kind of develop some quantifiable kind of results that we can see how well is it actually neutralizing. We can assume that antivenom obviously works and it saves lives, but how well is it actually working against these specific toxin families? And yeah, so that's kind of where we're going now. So we've been starting to do these in vitro, so just like in a test tube assays of antivenom against certain toxin families. So like we mentioned earlier, we have biochemical assays where we test the activity of a certain protein family. So I'll just take PLA2, for example, where we have our venom from a specific species, and then we mix that with some antivenom, and we allow that to sit for a certain period of time. And then afterwards, we conduct the assay as normal, and we see how that activity is different than without antivenom. And from those results, we are seeing a reduction in activity. However, not as much as we might want, and it's not very efficient either. So we did 2 concentrations. We first added 10 times the amount of antivenom as venom to these tubes, and we tested its activity. And there is a little bit of a drop. But we also did 100 times the amount of antivenom as the venom in the test tube. So that's quite a bit of antivenom. And as many people probably know, antivenom isn't cheap. So we see that there is obviously much more of a reduction in activity at that 100x concentration. However, there's still a significant amount of activity from multiple protein families that we've tested for. So for PLA2 and also these SVMPs, there's still a lot of activity for considering the amount of antivenom we've administered to these tubes.
Is there in theory, a theoretical way to make these antivenoms more efficient? So we need to use less of it to treat, obviously, like I said, they're expensive. They can be limited. The availability of them, even in the United States, can be limited. But if we needed to use less of them because they were much more efficient, we might have much more antivenom for the world. Is there any hope, or in theory, do you think we could build a more efficient antivenom?
Yeah, actually. So you've had both Dr. Stephen Mackessy and Sam Kerwin on here, and both of them were just published in a Nature journal talking about these nanobodies that are being developed. And these nanobodies are made specifically to combat certain toxin families. We are seeing the same results that we got with our antivenom at a 1 to 100 level, at a 1 to 10 level. And so there are absolutely developments in place happening right now, looking at reducing that in the future.
Gotcha. I remember that. I was just throwing you a softball. No, but I did want, because sometimes people don't listen to every episode. So if they listen to this episode, I want them to hear that because it is fascinating and groundbreaking.
Yeah, absolutely.
I think something good to say is that these antivenoms, so Crofev and Antivip, the ones we're looking at, they are beneficial to people. People absolutely do need these antivenoms.
Good point. Yeah.
However, they're just not as efficient as we might like. So they do work. Just want to preface that so people don't think that.
Yeah. These are
absolutely don't work at all.
Yeah.
Okay. Great. Well, so that's the part that you're currently working on, and that's Cameron, you're moving on, and we'll talk about where you're moving on to here shortly. But Ariana, will you continue to work on this and take it further as undergraduate research? It sounds like probably have at least a couple of years left. But where do you envision this undergrad research going next? What's the phase 3 or 4, or whatever it might be?
Yeah. So I am graduating actually exactly a year from now, so next December. And right now we plan on proving what we've worked on a little bit further. So like we were talking about, we ran those PLA2 assays, really looking at that one toxin family. We want to show this in multiple other toxin families with some of the other stuff that we have. So I definitely want to run those. And I think both of us are pretty set on wanting to write a journal about this and try and get it published somewhere because, as undergrads, that would be an amazing goal to accomplish. And so a lot of the focus is going to really go into finishing up the experiments we have planned, really sitting down and writing all of this, and then moving on forward, through graduation. And I, love to stay doing Venom research for the rest of my life, but I plan on applying to get my PhD in a different lab. I have a lot that I'm emailing right now. Do
you want to, if you're moving away from Venom research, are you going more like biochemical, medical, or are you staying kind of in ecology evolution? What are you thinking for your future directions here?
Yeah, I think the majority of what I'm really looking at is ecology focused, a lot of conservation. Honestly, I would be happy staying in looking at the proteomics, or even genetics, but I really do think that, ecology is the path that I'll be taking.
Excellent. How about you, Cameron? What's next? Like you said, you graduated. she said she's interested in finding a PhD in the next year or so and moving on. What are next steps for you?
Yeah, next steps, med school is kind of the plan.
Mm-hmm.
I'm kind of just taking some time off right now just to work, kind of just take a little break from school before journey of med school begins. But yeah, like I said before, this has really kind of changed my perspective of what I'm interested in and just kind of working with these toxins and dealing with them from a medical standpoint. And that's something that I still have a lot to learn about and would love to learn more about as I progress in my career.
Yeah.
But yeah, med school, I think next.
Yeah, there are many, I should say there aren't that many doctors in the world that have are very accomplished at snake bite treatment and all of that. So you've got a leg up on some of that compared to other people who are medical doctors. So, well, that's great. Okay, we've been going almost 50 minutes now. So, it's flown by, but it's fascinating. I'm glad that we're able to get some undergraduate perspective on here and have you guys talk about your research and how you ended up having that opportunity. But before we close this one out, I like to have all of my guests tell me a snake story. So we'll start with you, Cameron. Tell me your best snake story.
Well, relative to many other people on this podcast, I've only been working with snakes for at most 2 years now. So I don't really have the craziest snake story, but my first rattlesnake that I found will probably do. I was hiking with my girlfriend in Moab, and we were just kind of enjoying the sunset, walking around. And I was thinking to myself, rattlesnakes are probably around here. Like, I don't really know exactly where they're at, or like, I don't know where they're at around this area, but it kind of strikes me as a place where they could be found. And we were walking and we reached kind of our end point, started turning back, and I was still kind of looking underneath these shrubs that kind of were just in the whole area. And right before we get back on the main trail where we're going back to our campsite, there's a little concolor sitting underneath this shrub there. It's a little midget-faded rattlesnake. And I was just absolutely amazed that we were able to find one, because it was pretty well hidden underneath this shrub. And my first rattlesnake I ever found on my own. And I probably sat there for 30 minutes just looking at it, just kind of admiring it. And we got it to kind of get in that striking pose. So we got to kind of admire that as well. But yeah, really good. Really makes you excited to find more rattlesnakes in the wild in the future.
Yeah, that's a great story. What does your girlfriend think about rattlesnakes?
I mean, definitely not into them as I am, but I think she's not necessarily super scared by them, just kind of, they're interested. She's also kind of interested in just wildlife in general. So she also was able to kind of admire it and not just run away at the first thought. So
yeah, great. Well, before we move on to Ariana, you mentioned concolor, and we've done some work with them in the past, both in Colorado and Wyoming. Where do they fall out on your broad scale kind of description of snake venoms? Like, because I've always thought of them as fairly toxic venom, but that could be completely off base. Like, do you guys remember where they fall out?
Yeah, actually, we're looking at our poster we have right now, and we'll see if we can. We work with, since we have 39 species of rattlesnakes, or I think it's like 38 species. It's hard to keep track of.
Yeah, of course.
Exactly what venom phenotypes are involved with what, but we'll see if it doesn't take too long to see.
Right here.
Okay. Yeah, so it looks like it's kind of cool that we talked about that, the absence of myotoxin binding with our antivenom. And this species has a very intense phenotype of myotoxin in its venom. So it could potentially be problematic when someone is potentially bitten by one of these snakes. And yeah, some of them are just kind of definitely scarier than others working with these snakes. So
this is one of the species that these new advances could increase treatment for proportionally the most, because
yeah.
Interesting. Okay. Yeah, that was fortuitous. But anyways, so okay, great. How about you, Ariana? Do you have a good snake story for us?
Yeah. Also, like Cameron, being very new to the field, there are not a lot of stories to choose from.
Raising garter snakes since you were a baby.
Yes, I did keep a lot of them. But I think my favorite story was I just got back not too long ago from studying abroad in Botswana for about a month. And I got to work with the Mokolode Nature Reserve out there. And there's a whole section specifically focused on rescuing snakes, doing snake extractions, and doing a lot of education wise. And so, being out there all by myself, I took a taxi. It was like a 30-minute drive. So I took a little taxi. I got all the way out there because I didn't know they didn't have a phone number for me to call. I just showed up and I was like, hi, my name's Ariana. Can I work with you guys? Just to help with the snakes because this is incredible. And they were like, yeah, absolutely. And after working with them for a little while, I was able to hold, of course, with a tube, one of their really large black mambas.
Oh, wow.
And obviously, in all of Africa, really, black mambas are a really, really big deal. You could go up to just about anyone and they would have a story of someone they know or like 2 people away who has died from a black mamba bite.
Mm-hmm.
And so being able to work really closely with those animals and really get to see behavior wise, talk with the people about the antivenoms that they have there and their supplies, all of that was an incredible experience. And getting to hold one definitely made my blood pressure rise. It was pretty scary, but it was an incredible experience.
Yeah, it sounds like it. Great. Well, yeah, I enjoyed it. And, I thank you both so much for joining me on the podcast today.
Yeah, thank you so much for having us.
Thank you.
And I just wanted to thank the audience and tell everybody to remember snakes are animals too, and it's a privilege to see one in the wild.